Okay, thank you so much for attending this presentation. My name is Jordan Clemsen, and today I will be discussing this paper published in 2023, titled "The Lung Microbiome Regulates Brain Autoimmunity." Um,
Can you here so you
yeah.
and press OK? Because
Make it bigger, like here.
oh, yeah, it's smaller. Yeah, and okay, so we can see. No,
Oh.
don't touch this anymore. The blue, okay.
Blue, okay.
That's
So
it.
you can see it. Okay, cool. All right, thank you so much. And like I said, so there we go. Okay. So briefly before I go into the study, I'm going to summarize some of the kind of concepts and the background relevant to this paper, just so we can piece them all together. So essentially, this paper does go over a couple of different things. There's all the first thing is the concept of glia. So first thing I think is essential to know for this paper is there's different types. There are the vast and diverse functions as well. So there's astrocytes, which essentially function as mediating blood and supplying nutrients to the neurons themselves, because neurons are basically essentially just go all the way to G0 in the cell cycle. And then they stop there, and because they're in G0 their whole entire life, just generating action potentials, they don't actually have the needs to actually take care of themselves. So these astrocytes, essentially, in the photo right here, the people online, I'm pointing to the one that looks like it's hugging a blood vessel. They essentially feed the nutrients to the neurons themselves and supply it blood. So they essentially do the processes that a neuron doesn't have to do, because it's going to just be contributing all its time to.
What do you call G0?
G0, it's a, so in the cell cycle, there's G1, there's an S phase, and then there's a G2. And essentially your cells usually go through that cycle. They replicate and supply their organelles. And then they simply go to S phase when they synthesize their DNA. However, because neurons don't really divide and go through mitosis, they don't really complete the cycle. So they go into G0, which is like it's like a loop that you go outside of the loop, if that makes sense. So there's like, if you imagine a circle of a cell cycle, there's this other circle up here that's G0. So when I say G0, I mean that this cell essentially just kind of goes into hibernation essentially afterwards, instead of just completing and dividing nonstop, because neurons don't go through mitosis. So that's what I mean with G0. So, and because they're in G0, contributing all their efforts to the action potential themselves, they have astrocytes that basically take care of them and supply the blood flow. So that's that. That's the function of it. Most astrocytes. You have oligodendrocytes, which essentially supply the myelin sheath, which is for engineering capabilities. It applies the insulation for the neuron itself. Without the insulation, it can't really have salt-to-door conduction, which is essential for certain action potentials to actually be electrical signals to be fired throughout the brain itself. So these oligodendrocytes repair this myelin sheath. It's a photo of it. I guess they just kind of labeled a little bit right there to kind of apply the insulation. If you think about it, almost like a jumper cable that's applying the rubber outside of it. That makes sense. Radial glia essentially aid in cell differentiation and development as well. And then the essential ones that is discussed mostly in this paper, but you're going to see why I say that mostly, is microglia, which are essentially macrophages for the brain. These glia are capable of phagocytosis, which essentially means they engulf any pathogens or anything else. They also have a really crucial role in brain development, which is why this is so crucial, I think, seeing this connection. They embark in this function called synaptic pruning, which is essentially where synapses are recycled, and these microglia will phagocytize any old connections, so that new connections can be established in the brain. This pruning method is pretty crucial for development and of the brain, and it can also be among other things. So there's a little bit of controversy in glia, which is I think it's something to touch on. If you can't tell, These have a vast amount of functions for something that's just something as simple as a cell. It used to be, well, prior to this discovery that oligodendrocytes do actually fire at action potential when they're forming the myelin sheath. It used to be that neurons were defined as just having an action potential, and that was just their defining characteristic. So essentially, there's a revolution probably going on about this notion so far. This paper I think was published in 2022, so it's really relatively recent. And I think it's something to keep in mind while we're discussing when I discuss the microglia section of this paper. The paper also discusses T cells, which are essentially part of the adaptive immune system, which essentially means that it's it takes antigens created from foreign invaders, catalogs them, and almost learns from the environment as well. These T cells are generated from essentially just stem cells that can become in the bone marrow themselves. They are capable of they produce cytokines, which cytokines are essentially just any chemical that calls in other white blood cells or lymphocytes. So that's kind of important to know. I mean, it's kind of an umbrella term, whether it's a chemokine, whether it's a pro-inflammatory cytokine itself. It's just cytokines. It's vague. They can call in other cells that then as well do phagocytosis. I'll discuss a little bit more about those later when we get to the lung section. One thing to keep in mind is cytokines are part of an innate immunity, which just typically includes inflammation, which the innate immunity means that it's essentially in our genes, essentially. That most of these genes themselves can be distinguished from genetic factors. When I get into the paper, that will become much more clear, as opposed to T cells adapt more because they take the antigens from their environment. Okay, and then in the last section, before I get into the background of the study, as just a once over of lung immunology. So the lungs function essentially is to provide oxygenated blood. To your system, which is crucial for the brain because you want to have ATP to actually generate an action potential. This is where the energy comes from. The lung does this by creating these van der Waals bonds, which are really weak bonds. In the lung and the alveoli themselves, this is really crucial. There's a lot of them in the lung, so it takes in from the air and the oxygen itself. Because it's such a crucial environment, we're essentially just taking in air from the outside source. The lung is usually filled with neutrophils, which is the most abundant white blood cell. These neutrophils are capable of phagocytosis and other methods. There's also evidence there's a virome in the lung itself and the mucus surrounding your lung. So there's probably a lot going on there. It's a really complex field, but essentially you're taking in air from the outside. You're hoping to turn into oxygen so you can power the rest of your body. Typically, when things like this do fall apart, I think there's one more slide after this. Fibrosis can occur, which essentially is inflammation and scar tissue that happens in the lung due to the healing processes. This is a natural healing process found throughout the body, but because scar tissue is more dense than normal tissue, it will lead to a inability, it hinders the ability for your body to absorb the oxygen from the air due to the rigid formation of the lung, but also because of the fibers that develop from your lung actually healing itself, which then hinders over time the ability to absorb oxygen efficiently. And then really briefly, sorry, this is the last slide, I promise, is really briefly just to go into the complexity of the lung microbiome, because they do discuss this briefly. I'm going to use the example of influenza type A just to highlight how diverse this can be typically. So influenza type A is a really good example of this because it essentially usually affects in the lung tissues. It attaches to acidic receptors in lung cells itself, and essentially forms an endosome, which is essentially just like a little bubble when it can get inside the cell. It tricks the cell into replicating its DNA and then pops and the cycle repeats. Which is really interesting about this virus itself and why I chose this to explain this concept is because this virus adapts by tricking the rest of the lung into aiding it based on our defenses. So we have these things called goblet cells in our lungs, which produce mucus, which is essentially a defense mechanism that helps with fluid. These viruses are able to trick that signaling pathway into providing more mucus. So then the microbiome in the lung actually interacts. And other pro-inflammatory cytokines then are fired nonstop, and you end up with this thing called cytokine storm, which is a really big, complex issue of just lots of different autoimmune cells coming to the lung with the mucus involved as well. So if you can't tell, it's a really complex situation, which is why I think when I get into the PCA, why it was crucial for this paper. Okay, and then I did list the pro-inflammatory cytokines itself, tumor necrosis factor alpha, and just to summarize, they're really it's sometimes anti-inflammatory, but it's usually pro-inflammatory. The interleukin 6 itself, which will be discussed in this paper as well. Now that I briefly went over those concepts, I'm going to go into the study. I think the best way, just because they did a really good job of blocking for this study for other variables, just due to the complexity, I think the best way to highlight this was to students to review it sequentially. I have figures towards the end, but the figures themselves are final, if that makes sense. So I will review this afterwards if that helps. So the first thing the study did was they basically essentially wanted to manipulate the microbiome due to previous studies in antibiotics in the lung, influencing unboxing the lung, influencing the immune system and the microbial environment with what is it? And there's been previous literature that suggests that there is probably interaction in the brain because these cytokines actually have a similar pathway. So that's why it's kind of interest. Like interleukin 6 has actually been it's been neurodegenerative diseases like Huntington's disease and Castle. Notch, the gene itself, actually interacts with cell signaling itself, along with the epithelial growth factor itself. So I mean, alterations in that suggest there is an alteration in other autoimmune processes as well. So in order to manipulate the microbiome itself, they injected intratracheally a 1 milligram of neomycin into mice. These mice were then essentially monitored for their myelin basic protein T cells, and they were measured itself. These mice were then left alone essentially for 6 hours, and then immunized so the antigens were actually developed itself. They looked into the normal the experimental. They also had a control with a normal experimental autoimmune inflamed lung itself, and they looked into and so they can compare these. They measured the results from the group themselves, and I'll get into how they measured it in a bit because it's easier explained later. But they measured this, and then they did notice a little bit of separation. So then they and to block for other areas in the body. They repeated the same process, but then instead of injecting the neomycin into the intratracheally, they injected it into the gut itself, so they can block for the gut microbiome. So they're just measuring the lungs. Once they blocked for that, they found minimal changes themselves, and they applied tenfold more concentration just to rule out any other any effects of weakening the immune system, and then how that would occur with and how that would influence a more resistant microbiome. They also and then they also did extract fluorescent cells themselves into the T cells as well. And when they inserted these fluorescent T cells, they were able to track their whereabouts and how it did diffuse past the meninges. Okay, So what they did notice is there's a different T cells number in the lung versus the central nervous system itself. And so, which was odd because you would think that just because by altering the lung and it wouldn't affect the brain, but it didn't seem like they did. So what they did to clarify, they ran a qPCR on the cytokines itself, which are the genes that then express the genes that then code for the cytokines expressed. Which also then back up their results as well. So then they did more blocking, which was they also then just injected the neomycin into the skin as well, just to block for anything else, because we don't know if there's other microbiome interactions that were then influencing the lung and the brain itself, because that's two different organs. And then they didn't find it, and they found the results were still conclusive. So they discontinued the injections of neomycin and switched to injecting just the microbiome in these rats into other rats to see if it was just the microbiome itself, not just the immune response that was occurring. So they injected the bronchial alveolar lavage fluid itself into other rats, and they were able to find that the microbial transfer, when they did inject another bacteria, the microbiome into another bacteria, another one microbiome into another mouse, they were able to find that these actually did reduce the inflammation in the lung, which is interesting. What they found also was that the T cell count was actually reduced when they did just inject one microbiome into the other. And they also did notice that from the fluorescent cells, it did seem to migrate past the blood-brain barrier, which is really interesting. So to continue on with the study, they did this new technique called, it's not new, but flow cytometry, which is essentially you take some lasers and you shine through a cell, and the refraction of the light through the cells and the organelles can distinguish the different chemical compounds of any antibodies, and you end up with like almost like a variance of how different each cell is based on just the refractions of light, if that helps. So they
it feels like spectroscopy.
Yeah, it is actually. It's what is it? It's there's an acronym for it. It's like 2-photon laser spectroscopy. Yeah, it pretty much is. It's like flow cytometry because they like because you can track the antigens and see the differences and see
What's the flow means there?
the flow. That's essentially because you can see where the cytokines are going with the antigen. So like when you shoot the light onto the cells and you see there's certain refractions, you can see the difference in antigens and you can know where in the body they were going, because based on the that certain cells in one area are going to have an antigen, the other are not. So the flow is that. What they found was that there's not a significant regulation of the genes. So it wasn't genetic as well. Comparing the qPCR of the cytokines themselves. So it suggests that there's probably just an environmental factor itself. And they also did check to see if the meninges was leaking, because in previous literature with immunocompromised patients, such as HIV, there does seem to be a crossover between bacteria itself and the brain. Okay, so now that they kind of established that there was that it's the microbiome affecting the lung, and it's somehow translated to the brain, they use this flow cytometry to measure cytokines themselves. They found that there was reduced expression in certain cytokine levels, and these cytokine levels have been shown to decrease. They also did notice a clinical decrease in inflammation in the lung tissue itself, as well as the brain tissue itself. And they did use the flow cytometry and qPCR on the microglia themselves. They found that the genes were upregulated in the microglia. So, like, just to summarize, the lung immune response was just not quite the same, but the response downstream of this pathway showed an upregulation of these genes listed here. And then they also did find that these other genes were downregulated. So what they did after this, and this is where we get to where the computer science stuff is, they ran PCA on the fluid in the lungs because they could see the different types of bacteria. They looked at they use a Shannon essentially. They use Shannon, sorry, to differentiate between the bacteria themselves, so they can like see how diverse they are genetically. And then they also did run a ANOVA, which I will show in a bit, the figures as well. Their most significant results was the gram negative bactericides of the family, 37%, which is really interesting because this family itself has been shown to well, recently we had the Nobel Prize for micro RNA. These bacteria have been shown to generate their own microRNA that communicates. So this is the kind of one that we think about in the gut that then communicates with our nervous system as well. So that was kind of interesting as well. I think that's why they presented that. They also had other bacteria as well, but not just the bactericides, but there probably is some interaction going on there. They also did mention that some of the anaerobes are resistant to neomycin. So when you're treating the lung with neomycin, probably part of the reason it helps is because it's altering the diversity of the bacteria in the lung, and that's leading to more of these bactericides to be more dominant because they are resistant to the neomycin itself, which is probably why that treatment's helping. They also looked at the in the cell wall bacteria, some of the lipids in some of these gram-negative bacteria itself, the lipopolysaccharides, were actually more abundant. And so what they did next is they tried to look at the I'm getting ahead of myself here, is they tried to look at they tried to just inject the lipid polysaccharides itself and see how that altered. And I'll get to those results in a second. What's also really interesting about these lipid polysaccharides is they do penetrate the blood-brain barrier, which then explains why there's probably a communication going on between and an alteration in certain genes in the brain itself. Okay, first figure in this study. So can everybody see this actually when I think about it? I can never tell if it's large or not. Yeah. I wish they would make this. Okay, so left top side, I need to figure out the size of this at some point. The PCA on the microbiota comparison with the lung fluid, essentially, on the left on the top side, as you can tell.
Can you point to it with your mouse?
Yeah, me.
Or with your hand.
There. Can you see my mouse on there? You can't. Right there. This is essentially the summary of what they did when they injected the this is essentially the methods, this part right here, this little figure. They injected the neomycin in the lung, switched it out eventually, and then just injected the bacteria themselves. Oops, I'm gonna trip over this at some point. And so essentially what they did is they and then the right is the abundance of bacteria and its influence on the gene. Like I said, essentially the procedure and what they did. And then C is figure C is going to be the one-way ANOVA and the Grisky Walls test that they did, as opposed to that. Figure 2 is the effect of just the neomycin on the brain. So that was the first step that they did. And as you can see, it was kind of okay, but I think part of that now, knowing what we know now, is that that is mostly due to selecting from certain bacteria that influenced the positive results in the brain. The B is the T cell transcriptome, and you can tell that it's not changed with the neomycin itself. So suggesting that the neomycin itself is not treating the inflammation of the lung. And then going from left to right, you can see the differentiating expression, the cytokine expression from the qPCR, and then the clinical parameters they did as well. They figure F is the transfers of the microbiota when they injected the microbiome of, well, essentially some of the fluid into one mice into the other group. And then G is the flow cytometry and the kinetics of the T cells themselves. And figure H is highlighted just for the fluorescent cells, and you can see when they're migrating past the blood-brain barrier. Figure 3 is the from point A. Is the effect of neomycin on inflammation and the inflammasome as a whole, sorry. And figure B is the T response with neomycin, and then if you can see, figure T is the C is the two-tailed t-test, and then NOVA. Figure 4 is just reconstructions of the microglia, and if you can see the fluorescent cells in the gray matter itself. B is just global gene expression, so that's with the and then there's also global gene expression throughout the whole entire body. There's also compared to the microglia count in the spinal cord in comparison to the control, and these clinical features. I'm gonna hold on there just a couple seconds. And figure 5 is the average microbiota in the lung. So that's just essentially the norm. And let's see if I can minimize this. I think it's kind of blocking it. I think is there a way I can like minimize this top part? There we go. Okay. So you can see the different heat map of the families of the different phylum of bacteria and how they influence certain things. And the clinical features. Additionally, with the injection of the lipopolysaccharides from the cell wall, and the one-way ANOVA with multiple comparison tests, just to compare all these together. Okay, and then so essentially what they did is they hopefully proved or showcased that there was a connection between alterations in the lung microbiome, its complicated interaction with the immune system, and how just that interaction itself can also alter the expression of certain cytokines in the brain and with inflammation. This is crucial. This could be of interest to neuroimaging itself because essentially it does deal with white matter hypertensive inflammation of the brain, but it is also regulated with just in the lung itself. So without even touching the brain, we alter just the microbiome of the lung, and then it changes certain gene expressions in the brain. Thank you so much. Any other questions? Might have been time-wise.
No, you're doing well. We still have about 20 minutes. Yeah.
Shoot, okay.
You went really fast. Like, let's
Yes.
Yeah, let's so this seemed to me like a heavy methodological study, right? They injected everything everywhere in different combinations, and then they measured and things like that. But like, I don't like just like, what is there like a single strong conclusion there that, hey, like as you said, like it was very hard for me at least to extract what is the main finding. Alterations of neomycin. So injecting neomycin into the mice in the lung decreases the versatility of bacteria, leading to bacteriocytes to thrive, which then increases cytokine responses in the brain and treats lung inflammation. That's how I'd summarize it. Treats as in addresses this, or reduces, I should say, yeah. And bacteria is not the endogen, like not the native bacteria, right? They were killing everyone, right? Well, because these bacteria were anaerobes and resistant to certain conditions, these bacteria were able to survive. So treating with the antibiotic they chose, selected for just the bactericides, as opposed to a lot more bacteria. And so that influenced the environment in the lung, which then influenced the cytokine expression of the brain. That helps. Isn't it, so why is it significant? For example, isn't it that just a control feedback loop, right? It's a control mechanism. That's how brain react. Like responsibility of the brain is to take care of to control all of those things. So something happened in the lung, feedback loop, we try to correct for that. I mean,
Right.
the brain tries to, the neurons.
Right, but like there was a fluorescent, so it did show that there was a not only different cytokine expressions, but also a migration from the meninges itself due to the lipid polysaccharides. So that I think that's what was crucial there, is that it did influence cytokine expressions in the brain as well, which influence inflammation, but also would trigger microglial count increase. So I think that's why it's important for the brain. Again, they didn't touch the brain; they just altered the lung, and then in doing so, they were able to alter the brain pretty significantly without and reducing inflammation in the lung.
Yeah, I may be not understanding. It's like, well, when you do something, anything to the body, the brain changes because it has to react to that, right? But you're saying it's more than that. It's kind of the bacteria percolated into the brain, or do that commonly, or that's
So what's really interesting about this is so they're able to influence activities in the brain, but not influence otherwhere in the body. Remember, they did a subcutaneous injection into the blood as well. So that was just to see results all over. And I think when you compare it to the global gene expression itself, I think that showcased that it mostly affected the brain. I would say. Yeah, you could argue that there's probably some extent that there's going to be cytokines expressing elsewhere, but not previously thought to this extent. I think that's what's really the interesting part, is that they just altered the lung and it affected the brain, as opposed to just altering the brain itself and then affecting the lung. If that makes sense. Again, there was one thing I liked about this paper was just how they really well, that's how I liked and didn't like. I like that they really tried to block for a lot of different variables, because it's a really complex thing, but it does make it sequentially difficult. That's why I put the figures at the end personally, is because it's not like a linear relationship. There's a lot of different things going on, so sequentially it just made more sense to block in certain ways, whether it's the blood, whether it's a subcutaneous injection, whether it's just the transfer of the microbiome itself, whether it's just the neomycin, whether it's the gut bacteria influencing anything else too. So I do think that future studies would probably need to. I did like how they worded it in their paper, because they didn't claim that there was like a groundbreaking relationship between the brain. They oversimplified a little bit with the bacteria, but part of that is just because it's so complex. So making a bold claim like that would just be, it wouldn't be great. Well, it wouldn't be valid, I think. And so I do think for further studies would have to look into different reactions. When I was reading this personally, I thought the virome would need to be addressed. There are so many phages in the lung. I just described how influenza A influences the lung, and that's a lot, and that's complex, because we're dealing with cytokines, we're dealing with bacteria, we're dealing with mucus that increased, we're dealing with the behavior of the participant, because you're not going to be breathing that well. I do think that they would need to, future studies would have to look into other microbial environments, especially since there is a known virome of phages in the lung. Any other questions? Sorry.
everyone is lost, I think,
Sorry.
in the PCA part,
The
especially.
PCA part. So
I'm just joking.
they use the valve, which is it's just like, so what they do is it's a diagnostic procedure where they stick like a tube down to your lower respiratory in your lungs, and they basically just like hose it out, hence the word lavage from French. And so it's that fluid. So it's just like everything in there. And I think that's why they use PCA. is because then it's it's uncorrelates, right? So then you can really look at the primary components themselves. I think that's why it's doesn't well the results of it doesn't.
No, PCA, it uncorrelates your Gaussian.
But yeah, that's what I meant. Yeah, so then like I think that's why it's beneficial to that. However, I've seen other studies that have looked into the that looked into this in comparison to neurological features. I have not seen some with ICA, so I'm kind of excited to see what that. It's essentially since we have PCA in our pipeline as a precursor to the ICA, I do think it'll be interesting, especially since behavior must interact with somehow with these interactions. Our microbiome does transfer from person to person, so I would like to see that. So
Anyway, so why are you interested? Why this paper?
Why this paper
your
is
interest?
interesting?
Yeah,
I think it did a really good job blocking and solidifying what it did. I see a lot of these papers that I just feel like there's always some other variable I could think of, and based on their bold claims, it just doesn't seem to like. I feel like I can pick it apart real easily. This one is really controlled, and it to me solidifies cytokine expression and regulation of the brain. And I think it does a great job of taking a very complex, multifaceted interaction and pieces it together in a certain way it's helpful. In our other lab, we work with influenza A and cytokine storm and walking pneumonia. And that already is just such a complex interaction.
Have you read the Book of Why by Judea Pearl?
I haven't read that. Is it good?
Yeah, it is good. And it's like he explains really simply a situation or many situations where controlling for something actually introduces a confounding factor. And people mindlessly kind of, I mean, not necessarily here, but that's what he writes. Often people just, follow procedure and control for everything, but in fact, it's like, you can introduce, if you have independent variables for parents to something, and then you're controlling for this child, they become dependent, although they're independent, and like things like that. So it's interesting, like this, just to add that the story is very complicated, right? Like it's just, I don't know how to control for everything.
Maybe there will be an uncertainty principle, developed at some point. Yeah, because I mean, it does feel like you have to compare for a lot, and then adding human behavior on top of it probably will be even more messy. So, yeah.
Yeah, I know you want to look at this cytokines in HD. Do you have any idea how you're going to begin to address some of that?
I am in the process of developing that. I have thought. So one of the so we are looking at certain cytokines. We're looking at so the ones I'm interested in mostly are tumor necrosis factor alpha and interleukin 6, just because those pathways are they're really clean in comparison to the brain. But some of the behavior is going to be more difficult. Part of me hopes that it'll be somewhat clean, but I think that's something I'm going to have to deal with, to be honest, because I'm hoping that a large enough sample size hopefully would help clear it up. Maybe it'll make it noisy. Who knows? But yeah, that's something I'm hoping to look for. Hoping that turns out less noisy, especially with how diverse.
Thank you, Jordan, for presenting.